The last of the ordered solids that we'll consider in this module and indeed in this course are the molecular solids. To frame this conversation, let's first look at an observation that typifies the various solids that we've talked about. All of the ordered solids generally have high melting points. So with metals we have things like iron, calcium, and silver that have melting points that are above 1,000 degrees Kelvin. The ionic solids are also very high, magnesium oxide even above 3,000 Kelvin. Covalent network solids like silicon dioxide also can have high melting points. In this case, silicon dioxide is almost 2,000 Kelvin. But we also have ordered solids like ice that melts at 273 and carbon dioxide that sublimes at 195. So what is it that distinguishes these last two substances from the other ordered solids? That's really the question that we're going to try to answer in this video. So our learning goals are going to be the basic structure of molecular solids, how they are different from ionic, metallic, and covalent network solids, and why that difference causes these melting points to be quite low. So properties of molecular solids. Covalent molecules are held together by different types of intermolecular attractive forces. We've talked about this in the previous modules. They include dipole-dipole forces, hydrogen bonds, which are a strong form of dipole-dipole forces, and dispersion forces, which are very weak forces. So the strengths of these interactions basically determine the temperature at which phase changes occur. The higher the melting or boiling point, the stronger these forces are, and when we have low melting and boiling points, we have very weak intermolecular forces. So a microscopic picture of this might include the following. Molecular solids are actually held together by these weak intermolecular forces. So let's imagine we have a molecule located here, and we have another one of its kind brought in, and they're held together by intermolecular forces. Now a third molecule might come and be held together by intermolecular forces, and a fourth, and a fifth. And gradually we can build up a solid by adding more and more of these molecules to create a distributed ordered network of these solids. So what we're creating is basically an ordered solid out of distinct individual parts. But those parts now, while the parts themselves are held together by strong covalent bonds, they're held together with each other by weak intermolecular forces that hold them together. And now melting doesn't require us to break the strong covalent bonds as it does in a covalent network solid. It only requires for us to separate these molecules from one another by breaking the weak intermolecular forces that hold them. Let's look at acetone as an example. Acetone is held together primarily by dipole-dipole forces, and of course all things are held together by dispersion forces. So here's a molecule of acetone viewed from the side. So one of the methyl groups is pointing at us here on the screen. It has a partial positive charge on the carbon, a partial negative charge on the oxygen, typical of carbonyl bonds. So another acetone might come in and it would try to line up its partial negative charge with the partial positive charge on the other acetone, also on that side. Then we might add a couple more and a couple more. Then we might add another row of these, and another row, and another row. And you can get an idea here of how a solid might be built up out of acetone, but all of it's held together by these very weak dipole-dipole forces. So in fact, when we try to melt acetone, we don't have to heat it up very much. It melts at 179 Kelvin. And for those of us who've worked with it in the lab, it's a liquid at room temperature. So clearly it doesn't have very strong intermolecular forces. So this is the essence of molecular solids, that they are basically independent units, molecules that are held together by covalent bonds, and those independent units are held together by weak intermolecular forces. That's what typifies a molecular solid. Now let's take a look at the structure of ice, which is another one. Water is held together by hydrogen bonds, so that's the strongest kind of intermolecular force that we can have. It's directional and it's stronger than dipole-dipole forces. So if I have a water molecule, it can now form hydrogen bonds to other water molecules. And in fact, each water molecule can form a total of 4 hydrogen bonds. So it can form this extended network. And each of the units in this network form a tetrahedral array around sort of this reference water. So it's an ordered network of molecules that are held together by hydrogen bonds, and the fact that it's ordered makes ice an ordered solid. Here's what a model of it might look like. And I'll note that if you connect the adjacent oxygens together, it actually looks like the chair form of cyclohexane. In fact, this is the diamond crystal structure that's reflected by this particular version of ice. So you can see that some of these patterns are actually universal in nature. This diamond crystal structure, this chair form of cyclohexane, we're seeing this appear even in the solid form of water. But that doesn't necessarily remove the fact that it's very easy to move these molecules apart and melt the water by raising it to a temperature of 373 Kelvin. Now, how do we determine the structure of these molecular solids? I mean, many of them melt at very low temperatures. They're not easy to see. How do we do that? Well, the way we do that is we can do the same we take the same strategy that we can use for any substance that forms a molecular crystal, and it's a technique called X-ray crystallography. In essence, what this does is it bounces X-rays off the molecule, and we can analyze the scattering pattern of these X-rays, and it tells us something about the bond lengths, it tells us something about the bond angles, the torsion angles, and so forth. A limitation in this technique, though, is that the hydrogen atoms are too small to be effective scatterers of these X-rays, so the position of them must be inferred in other ways. We can apply this technique actually to very large molecules, including molecules as large as proteins or nucleic acids. So this, in fact, is the primary technique by which historically we've been able to learn about the structure of proteins. It's historically how we learned about the double helix structure of DNA and so forth. There's a very nice student video made of X-ray crystallography made by one of my former students as their video project for a class, and I've included it in the lesson, and I urge you to watch it. I think you'll learn a lot from her treatment of X-ray crystallography.